I hope you're doing well today. Today we are continuing our conversation in chapter 3 over bioenergetics. This is part 2. So in our previous conversation, we talked about the terminology behind bioenergetics. We also discussed the central role of adenosine triphosphate or ATP in muscular activity. And we talked about the 3 energy systems for exercise and really covered more of the phosphagen system or PCR and glycolytic system. Today we're going more into the oxidative system. The aerobic energy systems which rely on oxygen include aerobic glycolysis and beta oxidation of fat. So aerobic glycolysis is the continuation of glycolysis in the presence of oxygen. It occurs within the mitochondria and is an important pathway for energy production during endurance activities. Here's how it works. Glycolysis starts in the cytoplasm and involves the breakdown of glucose into pyruvate. During this process, 2 molecules of ATP are produced. In aerobic conditions, the pyruvate produced during glycolysis enters the mitochondria. It undergoes a series of reactions called pyruvate oxidation, where it's converted into acetyl-CoA, releasing carbon dioxide as a byproduct. Now, acetyl-CoA generated from pyruvate oxidation enters the citric acid, also known as the Krebs cycle, which occurs in the mitochondria. Here, acetyl-CoA is further oxidized, releasing more carbon dioxide and producing energy molecules like NADH and FADH squared. Now, NADH and FADH squared produced during the citric acid cycle donate their electrons to the electron transport chain located in the inner mitochondrial membrane. The ETC uses these electrons to create a proton gradient across the membrane, which drives the synthesis of ATP through oxidative phosphorylation. Oxygen acts as the final electron acceptor in this process, combining with hydrogen ions or protons to form water. Aerobic glycolysis provides a relatively efficient pathway for ATP production, generating a larger yield of ATP compared to anaerobic glycolysis. It is the main energy system used during moderate intensity endurance activities such as jogging, cycling, and swimming. Now, beta oxidation is the process by which fatty acids are broken down to produce ATP. It occurs within the mitochondria and is the primary energy source for low to moderate intensity aerobic exercise. And here's an overview of how it works. So the fatty acids stored in adipose tissue or released from dietary fats are activated in the cytoplasm by attaching coenzyme A or CoA to form acyl-CoA. Acyl-CoA is then transported into the mitochondria. Once it's inside the mitochondria, the acyl-CoA undergoes beta oxidation, a series of enzymatic reactions. During this process, the fatty acid chain is broken down into 2 carbon units, acetyl-CoA, through sequential oxidation. Each cycle of beta oxidation generates NADH, FADH squared, and acetyl-CoA. The citric acid cycle, or Krebs cycle, and oxidative phosphorylation. So acetyl-CoA enters the citric acid cycle, or Krebs cycle, where it is further oxidized to produce NADH, FADH squared, and ATP. The generated NADH and FADH squared then donate their electrons to the electron transport chain, which leads to ATP production through oxidative phosphorylation. Beta oxidation of fats is a highly efficient process for ATP production, as fats contain a greater number of carbon atoms compared to glucose. This makes them a rich source of energy during prolonged aerobic activities such as long-distance running, cycling, and other endurance exercises. Both aerobic glycolysis and beta oxidation of fats play important roles in providing sustained energy during aerobic exercise. The three processes involved in the oxidative or aerobic systems are glycolysis. So glycolysis is the first step in the breakdown of glucose, whether it occurs in an aerobic or anaerobic environment. In the context of the oxidative system, glycolysis refers to the initial breakdown of glucose to produce pyruvate. This process occurs in the cytoplasm of the cell and does not require oxygen. During glycolysis, glucose is converted into the two molecules of pyruvate and small amount of ATP and NADH, or nicotinamide adenine dinucleotide, are generated. The pyruvate produced from glycolysis can then enter the mitochondria for further processing in aerobic conditions. Now, the citric acid, again known as the Krebs cycle or tricarboxylic acid cycle, or TCA, is a series of chemical reactions that occur in the mitochondria. It is the central pathway of aerobic metabolism and serves as a bridge between glycolysis and the electron transport chain. The main purpose of the citric acid cycle is to oxidize acetyl-CoA, which is derived from various fuel sources such as glucose, fatty acids, and amino acids. During this cycle, acetyl-CoA is completely oxidized to produce carbon dioxide, generating energy-rich molecules such as NADH and FADH squared. The electron transport chain is a series of protein complexes embedded in the inner mitochondrial membrane. It is responsible for the final step of aerobic energy production, which is the conversion of the energy stored in NADH and FADH squared into ATP. So the NADH and FADH squared, which are generated in glycolysis, the citric acid cycle, and beta oxidation of fats, donate their electrons to the electron transport chain. As electrons pass through the chain, their energy is used to pump protons or hydrogen ions across the inner mitochondrial matrix through ATP synthase, drives the synthesis of ATP from ADP, or adenosine diphosphate, and inorganic phosphate, a process called oxidative phosphorylation. Together, these three processes, glycolysis, the citric acid, and electron transport chain with oxidative phosphorylation, make up the oxidative or aerobic systems. They are highly efficient in producing ATP from various fuel sources and are the primary energy pathway used during sustained aerobic activities. In terms of oxidative metabolism, carbohydrate oxidation has a higher power output, but a lower capacity than fat oxidation. This is one factor contributing to the decrease in power output with carbohydrate depletion during prolonged strenuous exercise. The large increases in ATP utilization and glycolysis, as well as the strong ion fluxes during such exercise, results in metabolic acidosis. After the exercise duration extends beyond approximately 1 minute, for example, as in an 800-meter track event, oxidative phosphorylation is the major ATP generating pathway, and intramuscular glycogen is the dominant fuel source. During events lasting several minutes to hours, the oxidative metabolism of carbohydrate and fat provides almost all of the ATP for contracting skeletal muscle. Even during marathon and triathlon events lasting 2 to 2.5 hours, there is a primary reliance on carbohydrate oxidation. The major intramuscular and extramuscular substrates are muscle glycogen, blood glucose derived from liver glycogenolysis and gluconeogenesis, and from the gut when carbohydrate is ingested, and fatty acids derived from both muscle, the intramuscular triglyceride, or IMTG, and adipose tissue triglyceride stores. Primary determinants of the relative contribution of these substrates to oxidative metabolism are exercise intensity and duration. Keep going back to that, right? So carbohydrate oxidation, particularly from muscle glycogen, dominates at higher exercise intensities, whereas fat oxidation is more important at lower intensities. Maximal rates of fat oxidation occur at 60 to 65% of VO2 max. Oxidation of muscle glycogen and fatty acids derived from intramuscular triglycerides is greatest during the early stages of exercise and declines as exercise duration is extended, coinciding with progressive increases in muscle glucose and fatty acid uptake and oxidation. Accompanying the increase in muscle glucose uptake is an increase in liver glucose output from both liver glycogenolysis and gluconeogenesis. With prolonged exercise, liver glucose output may fall below muscle glucose uptake, thus resulting in hypoglycemia that can be prevented by carbohydrate ingestion, which is the reason why you see with marathoners that they'll often have some type of carbohydrate replacement. An increase in adipose tissue lipolysis supports the progressive increase in plasma fatty acid uptake and oxidation, but because lipolysis exceeds uptake and oxidation, plasma fatty acid levels increase. Inhibition of adipose tissue lipolysis increases the reliance on both muscle glycogen and intramuscular triglyceride, but has little effect on muscle glucose uptake. The importance of that intramuscular triglyceride oxidation during exercise has been a matter of debate, and results in the literature may be influenced by differences in individuals' training status, sex, fiber type distribution, and resting intramuscular triglyceride stores. Nevertheless, it does appear to be an important fuel source during exercise in trained individuals. Despite activation of the oxidative pathways in skeletal muscle during exercise, accelerated rates of glycolysis result in the production of lactate, which accumulates in muscle and blood, particularly at high exercise intensities. Although lactate was considered, again, to be simply a metabolic waste product for many years now, it's now recognized as an important substrate for oxidative metabolism, gluconeogenesis, and muscle glycogenesis, and as a signaling molecule mediating exercise adaptations and inter-organ communication. Glycerol is released from circulation from contracting skeletal muscle and adipose tissue, as is alanine from muscle, and both can serve as liver gluconeogenic precursors during exercise. Exercise increases protein turnover during exercise, and although amino acids, notably the branched chain amino acids, can be oxidized by contracting skeletal muscle, the contribution to overall ATP production is low. Under conditions of low carbohydrate availability, the contribution from amino acid metabolism is increased, whereas endurance training results in decreased leucine oxidation. Of greater importance are the post-exercise increases in myofibrillar and mitochondrial protein synthesis that underpin the adaptations to acute and chronic endurance and resistance exercise. Of note, aerobic ATP production is also activated during very intense exercise, and 70 to 100% of the VO2 max can be reached in an all-out 30-second sprint, whereas very little aerobic energy is provided in the first 5 to 10 seconds. 50% of the energy contribution in the last 5 seconds of a 30-second sprint is aerobic. So during the transition from rest to intense exercise, the substrate for increased aerobic ATP production is also muscle glycogen, and a small amount of the produced pyruvate is transferred into the mitochondria, where it is used to produce acetyl-CoA and the reducing equivalent NADH in the pyruvate dehydrogenase, or PDH reaction. The PDH is also under covalent control, existing in an inactive form at rest and transitioning to an active form via calcium squared positive during exercise. The influence of the calcium with help from pyruvate keeps the appropriate amount of the enzyme in the active form, despite increases in acetyl-CoA that would normally inactivate the enzyme at rest. During endurance exercises at intensities below 100% VO2 max, aerobic ATP generation dominates. In this situation, there is time to mobilize fat and carbohydrate substrates from sources in the muscle as well as from the adipose tissue and liver. The muscles still rely on anaerobic energy for the initial 1 to 2 minutes, when transitioning from rest to an aerobic power output, but then aerobic metabolism dominates. To produce the required ATP, the respiratory or electron transport chain in the mitochondria requires the following substrates: reducing equivalents in the form of NADH and FADH squared, the free ADP, phosphagen, and the inorganic phosphagen and oxygen. The respiratory and cardiovascular systems ensure the delivery of O2 to contracting muscles and the byproducts of ATP utilization in the cytoplasm, which is ADP and that inorganic phosphate, are transported back into the mitochondria for ATP resynthesis. The process that moves ATP out of the mitochondria and ADP and PI and the inorganic phosphagen back into the mitochondria are being intensely studied and appear to be more heavily regulated than previously thought. In the presence of O2 and ADP and that inorganic phosphagen in the mitochondria, the increase in ADP concentration with exercise is believed to activate the respiratory chain to produce ATP. This is an example of the electron transport chain. The protein complexes that comprise the electron transport chain, or ETC, fuel the production of ATP, which again is the universal biochemical energy currency. The electron transport chain, or ETC, is a mitochondrial pathway in which electrons are transformed from 88 plus, or the nicotinamide adenosine dinucleotide. In the context of NAD, redox reactions are a key component of cellular creation. When NAD is converted to NADH, it gains two things. First, a charged hydrogen molecule, and next, two electrons. As electrons are negatively charged, the combination of the positively charged NAD and hydrogen coupled with two electrons effectively cancel each other out and neutralize the resulting NADH molecule. That's why the NADH does not have a positive sign next to it. The charge of a molecule informs how it interacts with other molecules. For example, NADH can't do what NAD positive does, and vice versa. With the help of oxidation reduction reactions, or redox, a protein gradient is created which causes phosphorylation of ADP. The electron transport, or ETC, is the major consumer of oxygen, and thus ETC passes electrons from NADH to FADH to protein complexes and mobile electron carriers. Okay, so now we'll talk a little bit about fat oxidation. Here we go. Fat oxidation refers to the process by which the body breaks down stored fat molecules and converts them into usable energy. It is a crucial component of energy metabolism, particularly during periods of prolonged exercise or low-intensity activities. The primary storage form of fat in the body is triglycerides, which are composed of three fatty acid molecules bound to a glycerol backbone. When energy demands increase, such as during physical activity or when the body requires additional fuel, fat cells release stored triglycerides into the bloodstream. The process of fat oxidation occurs in several stages. Lipolysis, so the triglycerides are broken down into free fatty acids and glycerol. This process catalyzed by an enzyme called lipase, which is primarily activated by hormonal signals such as increased levels of epinephrine, which is adrenaline, or norepinephrine. The free fatty acids released from fat cells are transported through the bloodstream bound to a protein called albumin. This allows them to reach various tissues in the body, such as skeletal muscles, where they can be utilized for energy. Before fatty acids can enter the mitochondria, the energy-producing organelles within cells must be activated. This step involves attaching a molecule called coenzyme A, or CoA, to the fatty acid, forming a compound known as acetyl-CoA. The acetyl-CoA molecule undergoes a series of chemical reactions known as beta oxidation within the mitochondria. In each cycle of beta oxidation, two carbon atoms are removed from the fatty acid chain. generating acetyl-CoA, a molecule that enters the citric acid cycle, also known as the Krebs cycle. So acetyl-CoA enters the citric acid cycle, a series of chemical reactions that further breaks down the molecules, releasing high-energy electrons. These electrons are then passed through the electron transport chain, a process that generates adenosine triphosphate, or ATP, the body's primary energy currency. Overall, fat oxidation provides a significant source of energy for the body, particularly during prolonged aerobic exercises or periods of caloric deficit. It is influenced by factors such as exercise intensity, duration, and individual fitness level. Regular physical activity and a balanced diet can enhance the body's ability to utilize efficiently those fat stores for energy, which can be beneficial for weight management and overall health. Okay, so here's the good part. The complete breakdown of one molecule of glucose through aerobic metabolism or the oxidative phosphorylation results in the production of a net total of 36 to 38 molecules of ATP or adenosine triphosphate. The actual yield may vary slightly depending on the cell type and the specific conditions, but for simplicity, we'll consider the average range. So with glycolysis itself, during glycolysis, what takes place in the cytoplasm, one molecule of glucose is converted into the 2 molecules of pyruvate. That process produces a net gain of 2 molecules of ATP and 2 molecules of NADH. However, glycolysis also requires an initial investment of 2 molecules of ATP, so the net ATP yield from glycolysis is 2 ATP. In the presence of oxygen, the 2 molecules of pyruvate produced in glycolysis enter the mitochondria, and each pyruvate is converted into acetyl-CoA through decarboxylation, releasing carbon dioxide as a byproduct. Acetyl-CoA then enters the citric acid cycle or Krebs cycle, which occurs in the mitochondrial matrix. For each molecule of glucose, 2 molecules of acetyl-CoA enter the citric acid cycle. Through a series of reactions, the citric acid cycle generates 3 molecules of NADH, 1 molecule of FADH squared, and 1 molecule of ATP through substrate level phosphorylation, and releases 2 molecules of carbon dioxide. So the net yield from the citric acid cycle is 2 ATP. The electron transport and oxidative phosphorylation, the NADH and FADH squared generated in glycolysis, pyruvate decarboxylation, and the citric acid cycle donate their electrons to the electron transport chain, or ETC, which is located in the inner mitochondrial membrane. As electrons pass through the ETC, they release energy which is used to pump protons, the hydrogen ions, across the membrane, creating an electrochemical gradient. The flow of protons back into the mitochondrial matrix through ATP synthase drives the synthesis of ATP through oxidative phosphorylation. The exact ATP yield from oxidative phosphorylation can vary, but on average, each NADH molecule generates about 2.5 to 3 molecules of ATP, while each FADH squared molecule generates about 1.5 to 2 molecules of ATP. So the net ATP yield from oxidative phosphorylation is approximately 28 to 32. Combining the ATP yields from glycolysis, the citric acid cycle, and oxidative phosphorylation, you get a total net ATP yield of approximately 32 to 36 ATP molecules per glucose molecule. It's important to note that this represents the theoretical maximum ATP yield from aerobic metabolism. In reality, factors such as mitochondrial efficiency, proton leak, and other considerations may reduce the actual ATP. So this just gives you a simplistic overview of aerobic metabolism and the output associated with it. Protein isn't a significant source of energy for most activities. It's considered to contribute minimally during short-term activities, but can contribute anywhere from 3% to up to 18% of the energy requirements during prolonged exercise. Protein is broken down into its building blocks of amino acids, which are converted into glucose via the process gluconeogenesis. From there, it follows the same fate as any pyruvate molecule. Nitrogen, which is a waste product of amino acid breakdown, eliminated through urea or ammonia. Protein oxidation is a process in bioenergetics that involves the breakdown of proteins to generate energy. It occurs when the body needs an additional source of energy beyond what is available from carbohydrates and fats. While proteins are not the body's preferred energy source, they can be metabolized and oxidized to produce ATP, the primary energy currency of cells. The process of protein oxidation involves several steps. Proteins are composed of amino acids, and proteolysis refers to the breakdown of proteins into their constituent amino acids. This process occurs primarily in the liver and muscle tissues. Proteolysis can be regulated by various factors, including hormones and cellular energy status. Once proteins are broken down into amino acids, deamination occurs where the amino group NaH squared is removed from the amino acids. Deamination results in the formation of ammonia and an organic acid. Ammonia can be toxic to the body, so it's converted into urea in the liver and eliminated through urine. The remaining carbon skeletons from amino acids can enter different metabolic pathways depending on their structure and the energy needs of the body. Some amino acids can directly enter the citric acid cycle or Krebs cycle as intermediates, while others can be converted into molecules that can be used for energy production. The carbon skeletons derived from amino acids can be converted into intermediates of glycolysis or the citric acid cycle. Through these pathways, the carbon molecules undergo oxidation and generate reducing equivalents in the form of NADH and FADH squared. The NADH and FADH squared then donate their electrons to the electron transport chain, or ETC, and the mitochondria, leading to the production of ATP through oxidative phosphorylation. It's important to note that protein oxidation is typically considered a secondary energy source. The body primarily relies on carbohydrates and fats for energy production. Protein oxidation usually occurs under conditions such as prolonged fasting, so you'll have muscle wasting, intense exercise, or during periods of energy deficit when carbohydrate and fat stores are depleted. The breakdown of proteins for energy is generally not desirable as it can lead to the loss of lean muscle mass and disruption of cellular functions. The body prefers to preserve proteins for their essential functions, such as enzymatic activity, structural support, and hormone synthesis. So to summarize, during exercise, the body utilizes different energy systems to meet the energy demands. The contribution of each energy system depends on the intensity and duration of the exercise. So you have the phosphagen system, which is an immediate energy system, and also known as the ATP PCR system that provides energy for high intensity, short duration activities. It relies on stored ATP and phosphocreatine in the muscle. When exercise begins, ATP is rapidly broken down to provide energy. However, ATP stores are limited and it can be quickly depleted. The phosphocreatine then donates its phosphate group to ADP, regenerating ATP. And this system can provide energy for activities that last approximately 10 to 15 seconds, such as a sprint or a heavy lift. The glycolytic system or anaerobic energy system also provides energy during high intensity exercise lasting from 30 seconds up to 2 minutes. It relies on the breakdown of stored glycogen, which is the storage form of glucose within the muscles or glucose from the bloodstream. And it converts glucose into ATP and lactate in the absence of oxygen. And it typically can generate energy rapidly, but is limited in its capacity and produces lactate as a product, which can lead to fatigue and a burning sensation in the muscles. The oxidative system, also known as aerobic metabolism, provides energy for low to moderate intensity exercises lasting more than a couple of minutes. It utilizes oxygen to metabolize carbohydrates, fats, and to a lesser extent proteins to produce ATP. Oxidative systems highly efficient and can generate a large amount of ATP. It becomes the predominant energy system during endurance activities such as long distance running, cycling, or swimming, and relies on the breakdown of glycogen stored in the muscles and liver, as well as fatty acids from adipose tissue or intramuscular triglycerides to sustain energy production. The relative contribution of each energy system during exercise varies depending on the intensity and duration of the activity. In short, high-intensity, short-duration activities rely heavily on the phosphagen system. Moderate intensity activities involve a mix of phosphagen and glycolytic systems, and long-duration, lower-intensity activities primarily depend on the oxidative system. It's important to note that all three energy systems are interrelated and work together to provide energy during exercise, albeit to different extents based on the specific demands of the activity. So bioenergetics is the study of energy flow and conversion within living organisms. It encompasses the processes by which cells and organisms acquire, store, and utilize energy to perform various biological functions. ATP is the primary energy currency of cells. It consists of adenine, ribose sugar, and 3 phosphate groups. The high energy bonds between the phosphate groups store potential energy. When ATP is hydrolyzed by the enzyme ATPase, it releases 1 phosphate group, resulting in ADP and inorganic phosphate, along with the release of energy that can be used for cellular processes. The 3 main energy systems that contribute to ATP are the phosphagen system, glycolytic, and oxidative system. And I feel like we've covered the rest of this primarily. The energy substrates, the carbohydrates, fats, and proteins serve as energy substrates for ATP production. And the body's oxygen consumption is an important measure of aerobic energy production. Oxygen is needed for the oxidative system to efficiently generate ATP through oxidative phosphorylation in the mitochondria. The maximal rate of oxygen consumption is also known as VO2 max and represents an individual's aerobic capacity. So efficiency of energy conversion varies across the different energy systems, with the oxidative system being more efficient and producing a higher yield of ATP per unit of substrate compared to the anaerobic systems. So understanding bioenergetics helps us to comprehend how energy is generated, stored, and utilized within living organisms. It provides insights into the metabolic processes underlying exercise, cellular function, and overall energy balance.